Hello? Morning. How are you? All right, let's get started. So welcome to the meeting today. This is our last meeting of the year for 2024. So hopefully we can go over some things, tie up some loose ends, move into 2025. That should be good. This week we had the DevoWorm meeting. If I go to our meeting archive for DevoWorm, which is right here, you can see I've prepared an archive on GitHub, and so this actually goes back to the beginning of 2020. So this is a long set of meetings, weekly, annotated, and it has a link to the YouTube content, and then some social media threads. And then every once in a while we take a break from DevoWorm and we have some meetings, and those meetings are listed just for people's edification to know kind of some of the other things that we're involved with aside from the meetings. So we start with January 2020, and last couple of weeks we've been doing some interesting things. You notice we cover a wide range of topics. We generally have people presenting in the meeting, but I usually take the lead, so I'm usually listed as the lead for most of these, but sometimes we get other people. We always get like good, some good topics, just kind of like this meeting. I see in December we had two meetings. Last week, which was the 9th, we talked about reverse engineering C. elegans and some of the things involved with that. I also, after the main meeting, I usually do sort of an in-depth deep dive into some set of papers. This time it was a paper on developmental and tissue networks, which was very interesting because we've been talking, we have this long-term arc in the meetings this year on applying networks to tissues and organoids and embryos and other types of biological systems at the cellular level. So that was last week. The reverse engineering of C. elegans was this paper from a group of people who are proposing to do this work on characterizing every neuron in the C. elegans connectome and then doing behavioral experiments to show sort of this full range of behavior that can be generated by that kind of connectome. It's a very interesting sort of topical area, very nice vision, but it's going to be hard to realize. So this is something that I've had conversations with a few people in the group of the very large group of people who would propose this idea, and kind of saying how OpenWorm might be a good fit for some of what they're doing. So it does have some tie-ins to OpenWorm, but it's largely going to be their own initiative. In that meeting, we also talked about input-output functions, which are these, that's the way they characterize the connectome relative to behavior. So if you have, say, a sensory input, that's an input. Then you have this network, and then you have an output function, which is the behavior. It could be sort of muscle movements, or it could be some sort of feeding behavior, reproductive behavior. So that's, and then they characterize that in terms of linear equations, differential equations, and so forth. The week before that, on December 2nd, sometimes we do some troubleshooting in the group. Sometimes we do some more informal stuff. Susan Crawford Young is doing this work with COMSOL, which is an engineering tool. It's an engineering software package where she's trying to implement biotensegrities. And biotensegrities are these ultra-stable structures. If you're familiar with Buckminster Fuller's work on buckyballs or on geodesic domes, that's kind of, those structures are tensegrity structures. They're hyper stable structures that you can load with stresses, and they have, these opposing sort of these opposing forces that keep the thing ultra stable. So this is what she's trying to do. It's very hard because they don't have a native set of. First of all, it doesn't handle soft materials very well, but it also doesn't have a native set of physics or algorithms for handling those kind of systems. So it's, we've been talking about how to do that. We also talked about this article on sort of the operating system of the cell, which in the article we covered was described as the ribosome. And they talked about the bacterial ribosomes, of which there are maybe 50,000 in a typical bacterial cell, those being sort of the operating system of the bacterial cell, because if you don't have ribosomes, you don't have proteins. Ribosomes dock messenger RNAs, and they make proteins. They translate things into amino acids, and then those amino acid chains make proteins. So, we kind of talked about, and they kind of proposed that if you do the back of the envelope calculations on all of the DNA and all of the proteins that go into ribosomes, that description is basically can fit on a thumb drive, a typical thumb drive. So that's what they mean by ribosomal OS. Then we also talked about a paper on founder cells and early lineages in C. elegans. Founder cells are the sort of the precursors of different sublineages in the lineage tree and development. So it might be like an origin cell for making a lot of cells that become muscle cells or intestinal cells or neurons or epithelial cells or some other type of cell, differentiated cell in the adult phenotype. And so it was a really interesting paper, again, on these early lineage tree lineages and the founder cells and how they their sort of gene expression and their sort of metabolic behavior. So this is, I did a count on the number of meetings we had in 2024. We had 43. I think we're going to have another one Monday, so that'll be 44. So we had 44 meetings of DevoWorm this year. We had, I think, maybe 45 or 46 meetings of Saturday morning NeuroSim. So that's going to be, and it's just interesting to count up how many meetings you've had over the year and to give a report. Now, as I said last week, I'm going to be doing an annual report. I want to do it as close to the new year as possible because I may be getting another publication go live before then, and I wanted to include that publication in the final count. But I'm getting the presentation ready and kind of going through, again, doing this accounting. It's always really enlightening to see where you've been, to see where you're going, maybe, and also to know how much work you've actually done, because you always underestimate, because you forget things. You say, oh yeah, what about this thing that we did back in February? And it slipped the mind. So this is a good exercise for getting these things out. Okay, so we have this document. This is something Jesse has kind of resuscitated, and I've put some things on for 2025. These are these different sort of looking towards submissions for next year. So this is the 3 that we have so far. We're always on the lookout. There's a channel in the Slack. It's this call for involvement and events deadlines channel, and we put all sorts of things in here. And I'm just taking things from this channel and putting it into the spreadsheet so we have sort of a more permanent home for it. And then, actually, that's not the right one. This is the channel. And so this is where, if you have things that you want to post, maybe I'll put some of these special issues in, although I don't know if people want to target them more specifically, we can. But anyways, those are in this channel, and I will try to put them in this spreadsheet as much as possible. So we have 3 so far. We have embodied intelligence. And so this is, of course, we do this annually. We've done this since 2021. So 2025 is coming up. the deadline for submitting abstracts is March 1st, and you have to register and submit the abstract at the same time. It's like one abstract per author, so as in years past, we'll try to sort of, coordinate this if people are interested in doing multiple submissions. If we don't want to do just one submission for the lab, that's fine too. I have a decent idea of what we might do, but I'm not sure yet. Or at least my submission that I'm going to put in. So the event is April 2 through 4th, and then, that's usually it's a virtual event. They usually have some pretty good speakers. It's worth checking out. The New York Celebration of Women in Computing, Jesse told me that this deadline has been extended, so, we'll fix that when we get the new deadline, but I think Jesse's working on something for that. It's some sort of, like he's done in years past, is have this ethics buffet where he's focused on different topics in the area of AI ethics or in computing ethics or something. And then he's gotten people together to give presentations. And so I've participated in that in the past virtually, and it was pretty good. It's always a nice kind of adds value, I think, for the students, because it's a lot of undergraduates and they're looking to sort of get into a research area. They're looking to get into professional development. And so, I guess the project management class that I've been teaching has been influential to some people there. I've also done a number of talks, and that's been influential as well. So it's nice to have an impact in that way. Anyways, we'll talk about that maybe later. And then the MetaScience 2025 conference. So this is in the UK. It's a live person conference, but they will actually be having a virtual pre-conference, which is, I guess, right before the conference, and I'm going to look into that more. Maybe we'll submit something to this. And the deadline for that is February 7th. So it's actually happening in June and July of the new year, but the deadline for submissions is much earlier. Okay, so aside from that, we have, and this is coming up sort of in the first part of the year, is Google Summer of Code projects. So I know that Morgan had asked me about how we go about submitting projects for Google Summer of Code. So the details on that are, we need some sort of write-up of the project by maybe about January 15th. So, I can put that on the list too. And I'll put this on here as different categories. And we can have multiple. I just want to have a list of projects maybe that we can pursue. Project descriptions. And I'll put a deadline of January 15th on that. And we'll kind of go from there. I think the deadline, I usually have to send these to INCF, so the deadline is somewhat flexible, but it's usually about the middle of February. So we need to have it probably by January 15th, so we can think about which ones are the strongest ones going forward. Because if you want to submit 2 or 3 different project ideas, then we can work out what that project should look like. That would be good. Because we do that a lot, every year I try to at least generate a couple, and then I go through and I find the strongest ones and put those in. So we'll probably be continuing some of the stuff we've done last year. That'll be high on the list, and then we'll have maybe, and I know Morgan wanted to do something with open source and maybe some of the NeuroLinux stuff that we've talked about this year. So that's maybe something we can explore further. I just need to have some sort of project description by like January 15th. And, if we want to meet over the holiday, we can do that privately, kind of in a more informal session than this. So something to think about.
Yeah, that would be great.
Yeah. Okay, so let's start out. I had a couple things to talk about. So this week was we had NeurIPS, which is of course ongoing, and I've not been keeping up too much with NeurIPS. I know they've had their workshops, and we had the workshops and the tutorials that we went over last week, and I haven't seen much outcome from that, but and then I know they've had a couple keynotes already, so let me go through maybe some of the things online that we've seen. So this is the NeurIPS 2024 site again. Now they're into the conference, so it's going through Sunday. So they've, I think, covered a lot of things already. Let's see, they have some blog entries from this year. A couple blog entries already. The first one from December 11th was documentary filmmaking at NeurIPS. There was this other thing on results of the NeurIPS 2024 experiment on the usefulness of large language models as an author checklist assistant for scientific papers. Then they had the announcing the NeurIPS 2024 Best Paper Awards, and then they had the NeurIPS 2024 experiment on improving the paper review assignment. That was yesterday. And of course, so there are a lot of practical problems in the field, like how do you assign papers to reviewers? And, some of these other things about automating author checklists and other things. I want to get into the Best Paper Award because I think that's probably the most summarizable thing that I can show you. So every year, of course, they have Best Paper Awards. With that, we are excited to share the news of the Best and Runner-Up Paper Awards this year. We had 5 groundbreaking papers, 4 main track, and 1 data sets and benchmark track. So they have the main tracks and then the specialized tracks that highlight, respectively, new autoregressive models for vision, new avenues for supervised learning using higher-order derivatives, improved training of large language models, and inference methods for text-to-image diffusion, and a novel diverse benchmark dataset for large language model alignment. So, yeah, there are a lot of interesting innovations going on here. So the best papers for the main track, the first one here is Visual Autoregressive Modeling, Scalable Image Generation via Next Scale Prediction. So this is where they're using visual autoregressive models, and these are models that iteratively predict the image at a next higher resolution rather than a different patch in the image following an arbitrary ordering. So this is the novel visual autoregressive model. The VAR model shows strong results in image generation, outperforming existing autoregressive models in efficiency and achieving competitive results with diffusion-based methods. So we've talked a bit about diffusion-based methods in recent meetings, but kind of, how do you implement that? How do you do this with some of the other methods? So they're just comparing this with standard diffusion-based methods. They're also comparing this with autoregressive models alone, and then they're finding that this method is better than both of them. At the core of this contribution lies an innovative multi-scale VQ-VAE implementation, which is, I don't know what VQ-VAE stands for. I guess something about visual autoregression. So they're doing this at multiple scales of resolution, basically. The overall quality of the paper presentation, experimental validation, and insights or scaling laws gives compelling reasons to experiment with this model. Okay, so that's the first one. The second one is stochastic Taylor derivative estimator, efficient amortization for arbitrary differential operators. So this is a tractable approach to train neural networks using supervision that incorporates higher order derivatives. So a lot of stuff that's like multi-scale or doing like higher order calculus or things like that. Interesting where they're kind of going with the field. Such problems arise when training physics-informed neural networks to fit certain PDEs. So these are differential equations, partial differential equations. So they're trying to use physics-informed neural networks to fit certain partial differential equation solutions, or at least models. Naive application of automatic differentiation rules are both inefficient and intractable in practice for higher order K and high dimensions D. So we have higher orders and higher dimensions. And so we need to kind of fit to those things. And so using automatic differentiation rules doesn't cut it. So this is where they're kind of coming in with their new method. While these costs can be mitigated independently, say for example for large K but for small d, or for large K but small d using subsampling, this paper proposes a method, the stochastic Taylor derivative estimator, or STDE, that can address both. This work opens up possibilities in scientific applications of neural networks, and more generally in supervising training of neural networks using higher order derivatives. So those are the two sort of best papers. And then they have runners up for the main track. So they have not all tokens are what you need for pre-training, which is where you basically filter pre-training data when training large language models. The method builds on the availability of a high-quality reference data set on which a reference language model was trained. The model was then used to assign a quality score for tokens that come from a larger pre-training corpus. Tokens whose scores of the highest rank are then used to guide the final large language model training, while the others are discarded. This ensures that the final large language model is trained in a higher quality data set that is well aligned with the reference data set. The second paper is guiding a diffusion model with a bad version of itself, which is interesting. So this paper proposes an alternative classifier for guidance, which is CFG, in the context of a text-to-image model. CFG is a guidance technique or a correction in diffusion trajectories that is extensively used by practitioners to obtain better prompt alignment and higher quality images. So this goes into the details of diffusion models and how they're implemented, and they're using this adversarial approach to sort of train the model better to correct the diffusion trajectory. However, because CFG uses an unconditional term that is independent from the text prompt, CFG has been empirically observed to reduce diversity of image generation. The paper proposes to replace CFG by autoguidance, which uses a noisier, less well-trained T2I diffusion model. This change leads to notable improvements in diversity and image quality. Okay, those are the runners-up. And then we have the best paper for the datasets and benchmark track. The PRISM alignment dataset, with participatory, representative, and individualized human feedback, reveals about the subjective multicultural alignment of large language models. So this is interesting. Alignment of large language models with human feedback is one of the most impactful research areas of today, with key challenges such as confounding by different preferences, values, or beliefs. This paper introduces the PRISM dataset, providing a unique perspective on human interactions with large language models. The authors collected data from 75 countries with diverse demographics and sourced both subjective and multicultural perspectives. Okay, so that we've got, sort of this diversity. We also have subjectivity and multicultural perspective, benchmarking over 20 current state-of-the-art models. The paper has high societal value and enables research on pluralism and disagreements in human-aided reinforcement learning. So that's good. Yeah, so that's sort of the papers, best paper awards from NeurIPS. And yeah, so I don't think there's much more to say about that other than, I think they were talking about being on Blue Sky during the conference. I haven't seen much on Blue Sky from them, but, they're kind of going through some of the main conference stuff. So, and I'm sure that those websites for the tutorials and workshops will also maybe have some videos on them. So we might want to check out what's coming out from that over the next week or so. Another thing I wanted to talk about is the Computational Psychiatry Conference. And I think Morgan posted this in one of the channels. This is happening this coming year. Tübingen, Germany, July 14th through 16th. So this is the annual conference. This is the 2025 version. Now, I don't think there's anything we can do virtually. We might submit something if someone wants to go to Germany and present on it. I don't know. But this is basically the website for the upcoming year's conference. And so they have the program is sort of already in place. This is actually from 2024 below. A 2025 conference will be added when it's available. So they have the 2024 program here. It's basically, speakers. They have a computational tutorial on the first day. They have a clinical tutorial on the first day. And they have keynotes, a symposium here where they have a limited number of speakers. They have the Symposium 2. So Symposium 2 from last year was Reinforcement Learning Models of Physiological States and Traits, or States versus Traits, looking at longitudinal and neural studies. The Symposium 2, or Symposium 3, was Computational Mechanisms of Anxiety, Transdiagnostic Effects on Interoception, Planning and Avoidance. And I think we talked about Ryan Smith doing this sort of active inference flavor, and he was at this session. And that looks like it. I think Symposium 1 was Improving Task Reliability for Computational Psychiatry Research. Another thing I did this week was I went through our Slack channel, and actually the stuff that I've archived on the Discord from the Slack channel. So this represents over a year and a half, maybe 18 months or so, of posts to computational psychiatry. And so I put together kind of a list of things here, which are the different topics. I put them into 3 categories, and the methods, which I put into 3 categories. And this is far from complete. This is just based on some of the things that were posted in the channels. So computational psychiatry, of course, we have sort of our own flavor. I know Morgan talked about this, but basically I've tried to group things into 3 categories for each topics and methods. So in terms of topics, for category 1, we have a couple things. We have sort of a grouping that includes epigenetic signatures of developmental adversity and genetic dependent brain signatures. The category 2 is network topologies, connectomes, spectral signatures, morphometry, continuous statistical frameworks, overlapping systems, sort of nervous and immune system interactions, and then neuro AI, which is a lot bigger than category 1. But I think category 1 would be kind of like genetics, epigenetics. Category 2 would be kind of like networks and spectra and sort of continual measurement. And then, of course, you have overlapping systems and the neuro AI part. That might actually be a category 3. So let me put that in there now just to make sure that I have that as a separate thing. So that, yeah, I took that off. And then category 4, which would be this one, includes brain age predictions and deviations. So this is where we're thinking about developmental trajectories and normative developmental trajectories and being able to yield information out of that. So we have brain age and predictions and deviations. We have individual differences, which would be summarized as personalization. We have this idea of the exposome, which is the environment. So, people have tried to quantify the environment, and you can think of it as an exposome, which is, then roughly equivalent to a genome or a proteome or whatever. Then we have genomics or genetic roadmaps of the brain. So that kind of maybe goes into category 1. We might just put that over in category 1. And I'm trying to trying to refine this as we go along. Computational neural development, which includes infants in changing environments. So this is where we look at like the influences of the environment or changing environments on development from a computational lens. So all these things have sort of a computational aspect to them. So I mean, people are studying these non-computationally. Sometimes they're using computational methods, but all these things are sort of through a computational lens, and then we'd like to bring them together in these categories. So the idea would be to kind of work out these different categories, give them kind of a name, and then if that's something that we want to work into a paper or into multiple papers, or kind of think about those categories independently and then maybe together, that's something we can do. But I just wanted to get the ball rolling to put these different categories together so we can kind of get things organized. Now that's topics, and then methods, because topics are kind of these areas that you want to study and you're looking for methods to study them. Sometimes you'll be doing implementing methods. Sometimes the methods are sort of their own thing, their own topics, and so you want to explore those and say, oh yeah, this is a method that people are using. So the methods are divided into 3 categories. The first one is sort of a combination of graphical and dynamical systems and maps. So we have graph representation learning. We have dynamical systems, which yield these pathways, which we talked about in, say, the normative trajectories of development. We have repetition maps of DSM-5 symptoms. That was a method that I saw in one of the posts. Predictive processing, of course. We talked about active inference, but that's kind of, maybe that kind of goes in this category. So we might say active inference and predictive processing. Bayesian modeling of behavior, and what we call temporal depth. And for that, we have this dissociation and the tame method in that area. So we have this area called temporal depth. That's where we have dissociation, and then this tame method proposed by Levin. And we've talked about that quite a bit. But putting those together into this sort of graphical dynamical systems mapping category, maybe with some active inference and predictive processing. In fact, that might actually be its own category. I don't know how well it fits into that area. We might do a category 2 for that. Okay, and then it's probably its own category anyways, because it's quite meaty. So yeah, and predictive processing being different from active inference, but having them kind of work in the same space. Category 3 is gamification and discovering with AI agents. Then I have deconstruction here, which I'm not sure what that means, but basically, this idea that we can do sort of learning and discovering putting things into games with agents and modeling things that way. So we can have the potential for human interaction and other things. Category 4 then is Evo-Devo Neuro, which is, they like to use these kind of compound prefixes in Evo-Devo that kind of evolved from Evo-Devo. So you have like Evo-Devo Neuro, Evo-Devo Neuro, Eco Evo-Devo, all these things that are quite frankly confusing, but they're kind of catchy. So Evo-Devo Neuro is the evolution of development of nervous systems, or the development of neuro. So this is, kind of like where you're combining a bunch of different things. Evolutionary neuroscience, developmental neuroscience, and then the intersection of those. Then in that same category, we have emotional regulation with neurodevelopmental disruptions, or what I guess was summarized in some of the stuff that was posted there is aberrant parenting. And then, of course, your genetics methods, which are GWAS and next-gen sequencing. So all those things, they kind of fit in this category of looking at change over time, regulation, looking at evolution, looking at these sort of disruptions in the trajectory of development, and then looking at the genetic basis for that. So we have all these kind of things in the category 4. Let's move on. So another thing I'd like to share here is this. Actually, this is kind of shifting from talking about computational psychiatry to some of the preprint issues in neuroscience and some of the topics. So, we've talked about open science in the last several weeks. This is from Andrew Brzezinski on Blue Sky, and he's talking about the preprint battle in neuroscience. So maybe if people want a review of this, preprints were actually highly accepted in physics and mathematics for years. And of course, a lot of the papers from NeurIPS are also put on archive, and that's kind of where they live, and that's where people cite them, and it's very much accepted to cite archive papers in those fields. In fields like biology and neuroscience, however, it's not really been the standard. We usually get things behind paywalls, especially like abstracts from conferences and things like that. And so it's just a cultural aspect. Like, we could talk about in physics where the archive was founded maybe 30 years ago, and they were kind of ahead of the curve in a lot of ways in terms of not only accepting preprints, but people putting out preprints and it being part of the culture. In biology and neuroscience, there's this sort of anti-culture of preprints. That is that preprints are not, they're viewed maybe suspiciously. Maybe people don't want to be scooped. Maybe they don't want to get, ahead of their skis in terms of what they're, saying about their data and so forth. So it's not so much, there may be some practical concerns, but it largely is a cultural issue. So this person is actually part of the motor lab, or there's like a motor super lab at Western University in Ontario, Canada, and he's one of the investigators there. And they put out this nice weekly digest of papers where they send out some good papers on motor control and sensory motor behavior. So that's where he's coming from. And he's talking about the preprint battle in neuroscience. So according to Andrew, the preprint battle in neuroscience has gradually been won. I think the next step will be more dramatic, a collapse in peer review resources to service each paper. So one of the things about preprints, of course, is it provides open access, it provides early access to work. It allows people to say, take a preprint, have people look at it, evaluate it, and then have different versions of it where you can improve the paper before reaching a terminal publication. That's one advantage of preprints. And, you may or may not have a terminal publication, but that's kind of the path that a lot of people envision for this. The other thing, though, is this peer review aspect. So people have experimented with open peer review where, you have a preprint, you put it out, people review the preprint, you get feedback, you change the paper accordingly, and then eventually you end up issuing sort of a final version or a published version. And this is roughly how the eLife model works. The eLife model is where you submit a paper to eLife, they put it up as sort of a preprint, they have open reviews which are published, then you have to satisfy those open reviews, then it becomes accepted, and then it becomes part of sort of the published stream of papers on eLife. Now, a lot of people frown upon that for various reasons, and there's been this contentious battle between people who think that that's not a good model and people who think it is. But in neuroscience, we're getting this preprint aspect of the cultural change out of the way, and now we need to focus on the peer review aspect. And peer review is also a cultural thing. I mean, we have this culture of peer review where we review papers. We think of it as like this, check on quality, a check on, sort of the verification of things. And as we talked about last week, sometimes that's not necessarily the case. That you can have people review a paper and it still be fraudulent because they don't, say, dig deeply into the data, or it's maybe 3 sets of eyes instead of 300. So that's where we are with this. So continuing with this thread, vast majorities of papers, good ones, are incremental. This is not disparaging, expanding on established concepts and using standard methods. So what he's saying here is that a lot of papers are, when we develop a paper, we usually start with an outline, kind of like what we did for computational psychiatry there, and then we fill in the outline. We flesh it out. If we're collecting data, we collect our data, and then we see where we are. And we keep working on the paper. And a lot of times we will submit the paper at a certain point and then get peer review feedback and then improve the paper and then put it out as a publication. Now, if we're not preprinting it, that process is it's opaque. It's not transparent. It's private. And maybe, at some stage it should be private. But at some point, we want to get it in front of people's eyes. We want to get some feedback. And because of this incremental aspect of developing papers, we need to be able to do this, maybe, get it in front of enough people so that we can, it improves the overall flow and tenor of the paper. So you're really kind of trying to develop concepts, develop ideas and experiments. And it's really hard to do without feedback. We usually get this informal feedback where, we pass it around with people in our lab and we say, is this a good idea? Do you have better ideas? But that, again, is a limited pool of people. So this is where preprints and open peer review can help. As a vast majority of papers are incremental, which means that they just kind of develop an idea and they don't start with gold. You have to find that gold through sort of this iterative process. It's worth keeping in mind that papers are read mostly by domain experts who, whether they reviewed it or not, will still have their own views anyways. These don't really need peer review. Sometimes, when you have people giving an opinion piece, maybe we don't want to peer review those things. Maybe we do. But, if you have an opinion, and a lot of people have opinions, and, that's not something you need to necessarily peer review because it's something that is not, maybe you need to develop the idea more, but that's, just something that happens with more normal feedback and discussion. Okay, so that's, we have this class of papers that are sort of incremental papers, data papers, theory papers. We have this other class of things that maybe are responses to that, which are opinion pieces. Maybe those don't need peer review, as opposed to the other things that maybe need peer review as developmental pieces. And then we have these other types of papers that are making big claims or whose claims prove to become influential. These need much more serious peer review than they are currently getting, and the people doing peer review of these papers need much more credit for their work. So there are these papers that are sort of like watershed papers where that influence the field much more than other papers because they're dealing with maybe much more, sort of concepts that are being clarified or introducing new concepts, or maybe there are things that are going to be cited quite a bit. Sometimes you don't know what those are, but sometimes you do. And if you're making big claims, if you're making like some novel claim about human evolution, or you're making some sort of big claim about, given like you get access to a big data set and you make some claims. I'm thinking of like ENCODE from like 10 years ago, or, something from a brand new instrument where you're really kind of getting data for the first time and you're making some claims. Those need maybe more intensive peer review. Those are places where instead of putting usual 3 people on it, or 3 people that you can get at the time, having a more formal system for getting feedback and incorporating it. So this is kind of the case for this open peer review, especially for these types of papers. But you can also use open peer review for these incremental types of papers as well. Now, the reason I'm bringing up this thread in part is because there is this Nature paper here in Nature Human Behavior, the Future of Academic Publishing. This says a bunch of authors on it, including Andrew Brzezinski, Patrick Minot is on this, and some other people. And so this is maybe one of these opinion pieces. And it says, academic publishing is the backbone of science dissemination, but is the current system fit for purpose? We asked a diverse group of scientists to comment on the future of publishing. They discuss systematic issues, challenges, and opportunities, and share their vision for the future. So I don't have the paper, I don't have access to it. So we won't talk about that in depth, but just to let you know that this paper exists and that this is what is referring to in this post. He actually has his contribution here, which is his position, which is eliminate peer review. The preprint revolution has largely been won. Victory is secured fast and free access to much of the scientific literature. In many labs, including my own, the preprint is what is generally consumed, and the eventual journal version yields little more than a passing scan and update to the citation manager. This new reality puts into sharp relief the nature of traditional peer review, an erratic and often perfunctory process that tends to improve papers without fundamentally altering them. So this is where he's talking about this sort of iterative improvement of papers. But what tends to happen is people will post a preprint and they'll post updates, and then kind of like the final version in a journal might have some minor changes from those ideas that were posted in a preprint. And so, people will just use the preprint because they can get access to it and because it's virtually indistinguishable from the final paper. So this speaks to how people sort of treat preprints, right? Like you get things that are sort of late stage development, and then you put it in on a preprint server, and then maybe you get feedback, and then maybe you kind of go for a formal publication. So, with a lot of these cases, people are kind of entering that stream late. Well, what's interesting is you could say enter the stream early. You could put together like a very, we might call it weak paper, but it's something that you might want to get people's feedback on. Maybe it's a very important set of concepts. Maybe it's like our view on computational psychiatry. We don't want to put it all out there and say, this is our view and take it or leave it. We want people to say, oh yeah, that's probably pretty interesting. This has maybe been done before. This is kind of trivial, etc., etc. Getting that feedback early might be actually quite useful for something like that, but maybe not so much for a paper that just basically analyzes the data. Okay, so we should consider the idea that most papers do not need traditional peer review. And in fact, if you go back through the history of science, maybe 100 years ago or maybe 200 years ago, peer review was not what it is today. It wasn't really viewed as a check on thing. Maybe it was viewed as more of a check on quality than maybe like a set of interactions, or maybe they viewed it as a set of interactions without being this sort of, check on quality or something. So it wasn't like they didn't have the kind of formal system we have today, but it was also maybe more realistic where you just had this, people would give it a yes or no. Maybe it was driven by the editor. And certainly Nature falls into that category where, they're basically the editor drives a lot of decisions. Okay, so, maybe most papers don't need traditional peer review. Maybe we've gotten to the point where we're just applying this peer review model to everything, and it doesn't necessarily need it. Okay, so this kind of copies what he talked about in the thread, not subjecting most papers to traditional peer review would save substantial resources, author and reviewer time, but also funder money. The savings could be invested in more science, but also more rigorous peer validation of preprints that introduces new ideas and approaches or begin to garner substantial influence. In practice, peer validation would mean that reviewing the claims but also reanalyzing the raw data, scrutinizing and rerunning the original code, and perhaps even running confirmatory experiments. So really, when we talk about peer review, when we're talking about quality checks, we're really talking about gatekeeping in the way it's currently done, as opposed to peer validation, which is where you actually go through and interact with the data in the paper at a very basic level, basically deconstructing the paper and reconstructing it, and then saying, yeah, I've been able to construct this paper. Now, there are people who do things like, there have been a lot of experiments with publishing models, such as executable papers. And what that means is that you can take a paper and you could execute it, say, in like an interactive notebook, a Jupyter notebook, and you can actually reproduce the paper in that way. So in this case, this would be kind of like more like peer validation, or enabling this kind of peer validation, where you get this, basically I give you the tools and I give you everything you need, and you can walk through the paper and execute the paper as you go through the paper, as reading the methods, understanding the methods, and then understanding the results, going to the notebook, going through the different kernels of the notebook and executing everything. And then everything makes sense. And if it doesn't make sense, then you can ask questions, or sometimes you see results and you don't know what it is. You ask a question, and then the description in the paper might be updated so that it makes more sense to the reader. Because a lot of times what we do is, we make a statement or we kind of present an idea, and it's not necessarily very clear what that idea is. And so refining the idea, refining the description, unpacking things that need to be unpacked, that's really where a lot of the value of peer review lies. Sometimes it's detecting fraud, and this method can, of course, unravel that. If you're deconstructing the paper, you're interacting with the data at a certain level. Sometimes it doesn't, of course, and that's where we need to kind of focus our energies too. But really kind of, making sure that peer review isn't just simply gatekeeping, that it's this sort of enterprise that gives us some insight. Okay, so funders and journals would have to incentivize peer validation because, although rare, the effort would be substantial. Preprints would also need to adapt to include open data and code from the outset, as validation could happen years later. So, we could do validation years later if we revisit a paper in 10 years and see how it looks compared to some new data that comes out, or put new data into the same model and run it and see if it's still valid, or, whatever. There are a lot of fun things we could do there. Such a shift from universal peer review to rare peer validation fits with a growing desire to change how scientific productivity and impact are evaluated. Peer validation would be an achievement for authors, showing that their work is robust and impactful. Peer validation would itself become a valued scientific contribution for reviewers. This is where, you get credit for being a peer reviewer. A lot of times in papers, they'll list the peer reviewers on the front page, like in the Frontiers journals. Sometimes you get credit through some of the altmetrics, but and sometimes people will write blog posts about it. Say, I reviewed this paper. Here's my take. A lot of times people will view peer review as confidential, but sometimes, people actually treat it as this sort of interactive process that, as long as it's not mean-spirited, can be very enriching. So this is just kind of rethinking this whole relationship between peer reviewers sort of giving feedback and the authors of papers, and how papers are published. So, we really, ideally, we need to shift in how papers are published and presented. So to some degree, the shift is starting with subjournals advertising the review process rather than the outcome by attaching reviewer comments and author rebuttals to the manuscripts they publish. But again, this is reliant on peer reviewers really giving a deep, thorough investigation of the paper, or kind of really good comments, and not like a superficial set of comments or a mean-spirited set of comments, which happens often. Then it kind of talks about, there's some discussion here from people kind of thinking about this problem Yeah, so I think the old model of peer review is kind of unsustainable, and we need to kind of think about a newer way to sort of go over papers and evaluate them. So that's all I'm going to talk about for that. Now I want to get into some papers. This is a paper that I think we've gotten. This came out in November. This is from the journal Neuroinformatics. This is on interdisciplinarity and collaborative training in neuroscience, insights from the Human Brain Project education program. So this is highlighting the Human Brain Project, and they have this educational arm that kind of grew out of their main project. So, it's kind of like where, a large funded project might have some outreach components. They might have some educational components. They might have an open source community, these sorts of things. So this is describing their training and education program that they've kind of developed alongside the Human Brain Project main focus, which is data and modeling. So this is where, we have a number of authors. I'm not familiar with any of these people, but they were involved in this effort. So the abstract reads, neuroscience education is challenged by rapidly evolving technology and the development of interdisciplinary approaches for brain research. The Human Brain Project, or HBP, education program aimed to address the need for interdisciplinary expertise in brain research by equipping a new generation of researchers with skills across neuroscience, medicine, and information technology. So this is where, we have the Human Brain Project. It's producing this new type of data. It's producing resources for neuroscience, but we need to have this sort of interdisciplinary expertise. And I've seen this throughout my career where you have these projects that are ambitious. They bring together computation and biology and, other areas. And you need to train people. You bring together experts from different areas. And sometimes they talk past one another. Sometimes they kind of don't understand the big picture. And so there's always this aspect of these kind of projects where they try to get people, they try to build training programs that are inherently interdisciplinary. So you can build not just expertise in one area or another, but build expertise to see this big picture. And I think it's been a mixed success. I think sometimes people, benefit more from some of the siloed thinking, some of the siloed expertise that's coming together. So if you're like a neuroscientist, you might find some niche neuroscience-oriented niche that is improved by the interactions with computer scientists, and vice versa. So if you're a computer scientist, you might find an area that really benefits from interacting with neuroscientists. And then other people benefit from this interdisciplinary view, which is to see the dialogue between the two fields and sort of pick up on themes in that interaction. So I don't think it's, I think there's this aspect of education that's really valuable in a lot of these kind of projects where you have this bigger conversation, you have this synthesis that's going on, between different fields. I don't think everyone really can, build upon that. I think it's certain types of sort of synthetic thinkers, maybe people who are more theoretically oriented, but it does benefit everyone. And so this is kind of the sort of the goal of what they're trying to do. So, really what they're trying to do in this educational program is equip a new generation of researchers with skills across neuroscience, medicine, and information technology. Over its 10-year duration, the program engaged over 1,300 experts and attracted more than 5,500 participants from various scientific disciplines in its blended learning curriculum, specialized schools, and workshops, along with events fostering dialogue among early career researchers. So there are a lot of ways in which they approach their educational mission. They have this blended learning, specialized schools and workshops, events fostering dialogue among early career researchers. This is very typical of a lot of the approaches that other projects have used, where they kind of host workshops they'll have like these sessions where they kind of get people together, they kind of talk about the key issues, how to drive them forward. You always focus on early career researchers because they're going to be putting their careers out there in front of this initiative. So the idea is that their whole career is influenced by this kind of interdisciplinarity, by this kind of, bold initiative. Key principles of the program's approach included fostering interdisciplinarity, adaptability to the evolving research landscape and infrastructure, and collaborative environment with a focus on empowering early career researchers. So this is a whole set of words here. The first is fostering interdisciplinarity. So again, that can happen in a number of ways, basically pulling disparate fields together, sometimes siloed fields, and kind of exploring the intersections between those. It might be like having a course on neuroinformatics, which is, of course, combining informatics, computer science with neuroscience. It could be, some sort of interaction between neuroscientists and computer scientists where, you need to learn, if you're a neuroscientist, you need to learn computational tools. If you're a computational person, you need to learn neuroscience. And that's, of course, the second part, which is adaptability to the evolving research landscape and infrastructure. Basically, when we have these new technologies come out, it could be next-gen sequencing. It could be, this sort of omics, any sort of omics. It could be, new computational tools. It could be new neuroscience tools, etc. That you are able to adapt to those. You can learn about these things. You have a space to learn about them, and so forth. And then a collaborative environment with a focus on empowering early career researchers. So this again, where you have this open collaboration, it's not siloed, it allows early career researchers to take full advantage. So they kind of describe this whole setup of their educational program, and then, eventually these programs end, so you have to sort of close things down in under the purview of the program, and you have to write a report to your funder, but then sometimes you keep some of these things going. So a lot of these projects will have like a educational arm where they'll try different things during the course of maybe a 10-year program. They'll take the best things that come out of that and they'll spin them off. And so this is where, you can do some great experimentation and figure out what works and what doesn't, and then spin off the most successful stuff. So they provide an analysis and in-depth view across a diverse range of educational formats and events. Our results show that the educational program achieved success in its wide geographic reach, the diversity of participants, and the establishment of transversal collaborations. So this is where, you get people from around the world. A lot of times it's because you have, different collaboration sites. Sometimes you engage in a strategy of doing virtual education and virtual outreach. You get people from different populations. You get people from different perspectives and areas. And then the establishment of transversal collaborations, where you get collaborations from different places in science, maybe people from the broader community, and you get them working together. And a lot of that revolves around education, because you can't really have a successful collaboration unless you have some educational aspect to it, where people are learning from each other and there's some engagement there. Building on these experiences and achievements, we describe how leveraging digital tools and platforms provides accessibility and highly specialized training, which can enhance existing education programs for the next generation of brain researchers working in decentralized European collaborative spaces. Finally, we present the lessons learned so that similar initiatives may improve upon our experience and incorporate our suggestions into their own program. So this is where they kind of go over this. A couple words about interdisciplinarity here. In this investigative pursuit, the fields of neuroscience and engineering, these are the two fields that they've defined in their project, have integrated on many levels over the last decades. resulting in deeply interdisciplinary research fields. This synergy has been fueled by rapidly evolving methods, a collection of methods and technologies that enable collection of large data sets, analysis on an unprecedented scale, and large-scale simulations, which include high-resolution recording techniques and high-performance computing. Some of these methods were not new as such and were widely used by other communities, but not yet applied for neuroscience research. So a number of fields have emerged from this sort of interaction, from this synergy. These include neuroengineering, neuroinformatics, and computational neuroscience. But these developments require educational and training efforts that equip both emerging talent and established scientists with the skills and mindsets to remain competitive at the forefront of discovery. So this is where, we have to approach this in different ways. While some universities and institutions offer courses combining computer science and neuroscience, there is a scarcity of reports on integrating computational approaches in neuroscience curricula. And the traditional mathematical requirements for neuroscience majors are often lacking in their ability to allow people to analyze big data sets and other things that are needed. So this is really kind of the idea here. You want to give people the skills they need. Sometimes those skills aren't in the current curriculum, so you need to expand the curriculum. But we can only learn so much. We only have so much time to achieve that curriculum. So we can't. If we needed to learn, if we wanted to get people in a training program to learn everything they needed, they'd be there 20 years, and we don't want that. We want them to learn things fairly quickly. We want to get them through a degree program quickly and get them out in practicing science instead of constantly in training. So you have to figure out ways to do that. And so what they're showing here is this figure that shows kind of the participant range, which means that they have these events that are both small and large. And then the level of specialization, which is where you have different types of resources for learning. So they have their resources here. So the first thing here is this Human Brain Project Education e-library, which contains materials produced at education events and other lectures and learning material. So this is where you have these sort of archived materials, things that can be accessed asynchronously, and they have around 700 videos. So this is a very large number of potential training events that people can visit and interact with. The second one is the Human Brain Project curriculum on interdisciplinary brain research. This is where you have blended learning courses, where you have online courses with related workshops, teaching core research concepts and complementary subjects to non-specialists. And this involves 6 courses and 17 workshops. So again, plenty of opportunities to interact. They had young researcher events, which introduced early career researchers to this EBRAINS focus, and then focused on communities beyond the Human Brain Project. And for that, they had 7 distinct events that were hosted. They also had 7 student conferences, which allowed early career researchers to collaborate and exchange ideas. This Human Brain Project School, which were a series of advanced lecture-based courses with additional training sessions for certain techniques and soft skills. That was 8 events. The eBrains Workshop, which was this eBrains initiative, multi-day workshops to introduce tools and services available via the eBrains research infrastructure. That was 5 events. And then the eBrains infrastructure training events, which allowed us to get access to this research infrastructure. That's 35 events. So you can see it as sort of an initiative that you can structure things in a way that maximizes the number of sort of interactions. And sometimes these things are asynchronous and maybe background, and sometimes they're things that are more suited to these more intense interactions where you're engaging in a classroom setting or a conference setting or some sort of social setting. I just wanted to go, I thought that was an interesting thing to talk about in light of some of the open research, open science stuff we've been talking about recently. and how to structure sort of an interdisciplinary training program around those things.
Yeah, I mean, that's interesting too, just thinking about it in terms of what role like NeuroTechX has and what resources are kind of there for the people that NeuroTechX is bringing in.
Yeah.
That's kind of interesting.
Yeah. So
Yeah.
do they have like a YouTube channel, or do they have like courses, or do they have, what are their resources now?
In terms of eBrain?
Yeah.
Yeah, I mean, all of those things, right? I mean, I probably haven't spent as much time on them as I did when it was, the Human Brain Project.
Yeah.
Or when it had that branding. But I believe almost everything has been folded into that. So, and there's still, Karen Ammons, and I mean, she's certainly the face of EBRAINS as the director. She's giving talks. I think they still, I mean, they probably are having webinars that are focusing on the particular platforms that they have. But I mean, there's definitely, I wonder how much in this new iteration there's been less emphasis on the, I don't know what you want to call it, but like, basically the neuromorphic computing, the neurorobotic extra activities, or, like, I don't want to say extra, that sounds wrong. How much have they gotten past kind of brain imaging and brain modeling?
Right.
And which was definitely, I wanted to say, I mean, as soon as you brought this up, I wanted to point out that Henry Markham, is it Henry? Yeah. Has I think it's the 20th anniversary of Blue Brain Project.
Okay.
And and they're he's definitely doing a kind of retrospective covering all of their, obviously what he wants to call successes. We can certainly say there's been a lot of outputs, and yeah, but right. I mean, and like a lot of, certainly a lot of the kind of computational neuroscience that was Human Brain Project was very much Blue Brain.
Yeah.
Or I like you said, they're talking about just the extension of projects past perhaps their original funding or something like that. Like, Blue Brain's still cranking out software packages and releases, and I mean, I'm sure he's brought in new funding and things like that, but they had a lot to build on. But I'm also just trying to think, I mean, I don't know if we're going to make this particular advancing informal STEM learning grant. I think the biolab is going to be involved in something that's being submitted, but in terms of like NeuroTechX and things like, how much, I'm interested. INCF does a lot, right?
Yeah.
To beyond, they do some kind of community building, or certainly like trying to roll out kind of platforms for people. I'm just trying to think of projects that have been successful in terms of not only getting an output, but also kind of creating community support for it, and kind of like I wouldn't say an ecosystem, but like like real training program. I for instance, I really love the NeuroPixels workshop from UCL.
Oh yeah.
And it's a recurring thing. Great lineup of speakers to make all the talks available. You can really give yourself a pretty comprehensive education from all the kind of the resources that they help curate and publish for you. You know what I mean?
Yeah.
Like I don't see. I think it's one of the things that like I'm kind of disappointed that Blue Brain, for instance, doesn't do. Or again, I might not be aware of it, but like there's not as much emphasis on the how to get these tools better integrated into other people's work, and especially kind of what seemed to be the focus of that paper was like for getting early career researchers using these tools or using these technologies, right? Again, it's I don't want to say other people should be doing more work. I mean, just saying, like, in terms of thinking about like the effectiveness of open source software being more than just I've got it up on GitHub.
Yeah.
And again, like I think some of the criticism to Human Brain Project was, like you've got these platforms, but there's not enough kind of ramping people up into getting more people than the people that developed them using them.
Yeah.
But again, this is, I've perhaps got an outsider's perspective, and I know that they were certainly trying to collect metrics and things like that.
Yeah, the metrics are kind of interesting because you have these alt metrics which allow you to measure things that are not like publications necessarily. So it's like how many people are hitting our GitHub repository. We put something up on like an archive like Dryad, which is like open data. How many people are hitting our open data set? You have these kind of rough measurements, and it's like hard to know who's actually engaged in these sorts of things. So if someone looks at your open data set, how much are they engaging in it? How much are they using it? What are they using? Same thing with the GitHub repository. Are they really, they fork it? Are they really adopting it, or are they just kind of looking at it? And and it's hard to get like facilitate all of that and get really good, not just data, but like a good sort of assessment of how successful you are.
Yeah. Yeah, it's a good point. I mean, and you know, I hadn't commented on your earlier kind of section, but you know, again, like, yeah, what are the incentives around publications?
Oh yeah.
You know, is a really interesting question, and you know, I do think that there's you know, there's been just a kind of a backlash to you know, like an earlier time when kind of journals had a purpose
Right.
as a gatekeeper. Like only so much could be published, and obviously that's just not true anymore. But that there became this, you know, that the incentives to get published became, you know, overriding, say, the, you know, what everybody found, which was reproducibility,
Yeah.
and you know, and again, perhaps even, you know, the directions and the you know, it kind of begged a certainly positive results, and perhaps even a kind of overly sophisticated approaches, because again, you know, you had to pass this gauntlet. But anyway, I'm much more interested in terms of the kind of INCF questions, and like what's good for fostering collaboration and fostering more well, and fostering, you know, multidisciplinary collaboration, you know.
Yeah.
And you know how, yeah, what gets people over kind of the activation energy that is required for that kind of, you know, that kind of you know, what is probably most appropriate is kind of multi-investigator collaboration.
Yeah. Yeah. It's always the key, getting that, like sustaining it, not just like, oh, that's interesting, I learned something.
Yeah. Yeah. No, that's interesting to see that paper.
Yeah. Yeah, I think with publishing too, like, you know, we can publish anything. We can put things out. Like, it's the barrier to publishing is lower. Like, we can get things formatted and stored. Like, when I was talking with the Active Inference Institute, one thing they're really interested in, or that piqued their interest, was overlay journals, where you create, like, you have an archive of papers, and then you create a skin that kind of curates everything. And you know, the journal at that point is largely a curation function. So, like, you can have peer review and you can add that to it, but the curation is the key. How is it presented? How do we get this into people's hands so that they understand, like, what the significance of each paper is, that they can find it? And that brings up the point that really the constraint now isn't like it may be in like 200 years ago, where you would print things and get them to people. You know, so you might have people needing to get a copy, say, of Origin of the Species. And like in Germany, you know, Origin of the Species wasn't translated into German until decades later. And so then as a result, you know, that scientific community, you know, kind of had their own sort of developmental trajectory with respect to that work. I mean, and we had, you know, publications like that up until very recently, where there's this gatekeeping function, you know, where you're seeing certain papers, you're not seeing other works. But of course, now we can digitally publish a lot of things, and sometimes those things aren't great, but you know, but the thing is attention. You know, that's the constraint now, and I think, you know, like, from how people, you know, constantly complain that there's so many papers that they're overwhelmed, and you can use, you know, you can use like, you know, bibliographic tools, bibliometric tools, to sort of sort through all the papers out there to see the most relevant ones. But it really is a matter of attention now, more than like storage or transmission. So that changes things, because now, it's like, how do I get people's attention? Is it going to be through some prestigious, maybe more prestigious outlet? Maybe is it that people go to like nature.com and they see what the latest papers are and that's what they pay attention to? Or is it like some tool for like preprints? So like the archive, of course, is notorious for having these dumps of papers in different categories, and you have to sort through them and figure out which ones are most significant. Or do you rely on some, set of archival filtering expertise sort of ways of finding the best papers? Like I talked about how the Sensory Motor Super Lab has their email list where they kind of curate the papers and they say, this is what we're reading. And, wouldn't it be great if you were reading it too? But, so those are I think that's the constraint. And it's actually a little bit harder because that means that peer review is different. It's about, it's about quality control maybe. It's not so much about gatekeeping, but it's about getting attention on things. And that's different. That's a different set of things that we have to worry about. We don't want things that are clickbait, like that's basically how the internet's handled attention, and that's not what we want. We want something more productive.
Yeah, I mean, I thought it was interesting talking to Conrad Cordy about why he was so interested in to some degree kind of moving into a kind of new area for him. And I think it's interesting to think about how much, there was this time Tim Behrens was kind of called out for the fact that his lab had kind of given each of the brain areas had been associated with a PI.
Oh, yeah.
And was just like, this PI is the, amygdala person, and this PI is the, whatever part of the thalamus. And leaving aside, the reasons that he was called out for that, the main thing being that, like, at the same time, there is a certain, you get, funding in an area, you get started in an area, that becomes the papers that you kind of need to read, right?
Yeah.
And it does, when we talk about researchers becoming a little, I don't think siloed is the right term, but needing kind of attentional blinders.
Yeah.
In a somewhat justified way. In that, like this is the report that they're going to need to write in 3 years or 5 years or 1 year, right? And so it's being able to open your searches to the internet is something is kind of a luxury that a lot of people don't have.
Yeah.
So I like what you're saying about like there is something about being a gatekeeper when publications and certainly the kind of dissemination of them involved logistics. but there is something certainly to be said for you kind of have to be in the right email groups, or my advisor would say all the time that like being an editor of NeurIMS was being like, I forget if he was like not editor, but but being a person as part of a journal especially kind of a premier journal for a particular field, that was his way of knowing what he should be reading,
Yeah.
right? And so discuss, finding reviewers for things, deciding, what was good, what wasn't, or what were things that they were going to be suggesting, and things like that was, a real way to, steer the conversation in kind of a journalistic way, right? And yeah, it is kind of it is, I certainly know the problem where you go to meetings and not everybody's read the same papers.
Yeah.
And there's something cool to that, in the sense of like, it's great to kind of spread the word, but it's also like we have to meet again where you've read those papers, and now we can actually have a proper discussion.
Yeah.
And anyway, I'm sure you got more stuff, so I don't want to bog you down here, but these are really interesting. These are really it's in trying to think about what role Neurotech has. It's interesting in that, I think one of the first things that I have to address is like, who is our real audience? Like, who are we helping? And I think our lack of analytics in terms of, do students engage more? Is it programmers? Is it, because, I feel like we really need to tailor what we can only do so much. And then we so we need to absolutely be kind of like a journal, in the sense of, we need to know what we should be curating.
Yeah.
Eric.
Yeah, that's a common thing we found in OpenWorm. I used to be on the community committee there, and we thought about this problem, and we never really solved it, of course. But so I mean, one of the things there is that you get people who are interested in, like, well, a lot of people who are like computational people, might be software engineers or programmers or something, and they come into this world where there's like this aspect of biology, aspect of neuroscience, aspect of computational stuff, and they want to find a place, and then there are other people who are biologists who want to know more about the computational side. So you get these skill sets that people are kind of entering in with. And then it's kind of the job of that community or the people running the initiative to sort of balance it out. Like, say, if you're, you want to work on a problem, here's what you need to know. You need to get up to speed on maybe some of the aspects of the biology, but you don't want to treat it like you're teaching them biology in a biology course, because it's not going to be the same thing they're looking for, because they're ultimately applying it in a different way. So you want to figure out kind of what captures their attention, and sometimes you don't know what that is. It's kind of like what might capture a biology student's attention isn't the same thing that would capture an engineering student's attention, and vice versa. And there are things that they pick up, cues that they pick up on, that are sort of connected to what they've been doing previously that resonate with them. So that's one thing. And then, trying to collect data on this is like, we never really got beyond the questionnaire stage. Like, you might ask people what they're interested in, but you only know, they can only tell you what you ask them. So it's like, okay, well, that is something you need to work out more, and it's tricky. It's not easy to do. Yeah. So I mean, it's really, I think, really thinking about this at multiple levels. Like, what kinds of resources can we make for, different kinds of things that people maybe want to do in the community? Do they want to, learn maybe primarily be educated on certain things, but they want to work on a project where they have very applied interests. They want to sort of just hang back and absorb the community, and that's a valid thing, and it requires different sets of things to present to them, like
Yeah.
Maybe like recaps of what people are talking about, or bigger questions, or something like that.
Mm. Mm. Yeah. 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 Well, it's getting that feedback. I have newfound respect for how hard it is to get that feedback.
Yeah. Yeah.
Yeah. Yeah. It's kind of like getting students to give you meaningful evaluations at the end of the semester.
Oh yeah.
And not just complaining that, like, yeah. I'm gonna check that out, and yeah, it's worth digging into EBRAINS a little bit.
Yeah,
Or,
definitely.
I mean, the other thing that I'm kind of looking for is how much I say, like, Blue Brain's not doing enough.
Yeah.
That's not true. But they're not doing enough of this kind of work. Like, is that a niche that would be worthwhile filling?
Right.
Or because I've been kind of I was really. I think I mentioned this when we covered Montreal AI and Neuroscience, the main meeting.
Yeah.
I noticed that the kind of tutorials that they had, or I forget, the workshops associated, I think it was tutorials. They had this scikit-learn emphasis.
Yeah.
And which I thought was interesting for for kind of like one of the premier neuro AI conferences, right? Why weren't they doing one of the fancy pants things? But I'm really interested to know, because I still see that as, if you were gonna actually do some work in the real world.
Right.
like that's kind of like the framework you should probably go to first.
Yeah.
And get to know your data set. Like, you don't get to know your data set with PyTorch, right?
Yeah.
You get to know your data set with stats and, visualizations and things like that, right?
Right.
and that there was some good overlap between this NeuroTech project Moab and what Maine was doing, right? And that if we did a little bit of work in terms of just trying to adopt some of their tutorials, like, that would be a really nice it'd be a nice kind of on-ramp into this project.
Yeah.
And just how many other kind of opportunities like that are there? Anyway, I'm not adding anything at this point. But,
okay.
Well, something to follow up on.
Yeah.
And I've been using, I don't know what you maybe created the channel for necessarily, but the pedagogy channel, it is like,
that's where we got that paper. That's where I got that paper from. Yeah, you posted it there. November 7th.
No, that's good. I mean, but it's it's like I have been using that as a place to put, when I see people who are trying to do something in that regard. Well, trying to do something new, or trying to think about something.
Yeah.
Sorry, I didn't. Maybe I missed that when you introduced the paper.
Oh well, yeah. I don't remember. I don't think I said where I got it, but yeah, it was posted in that channel. So, and then there's some other things in here as well. But yeah, we'll have to kind of think about some of these alternative models, because I think they're useful for like open source education. How do you get people in the on ramp, as you say?
Yeah.
the projects and that sort of thing.
Yeah. I mean, I've been thinking about it. I'm kind of liking some of the things that are posted here, like this Artem Kirsanov. He's now a researcher at NYU, right?
Yeah,
Center for Null Computation. And again, okay, that's awesome, because his videos, we don't need to kind of replicate his videos. We just need to kind of curate them and see how we can support people with, say, some of the materials that he's already listed in his YouTube descriptions. Like there's already this great amount of material that just kind of needs to be better organized for people.
Yeah.
I forget what the alpha. How would you say this? Alpha Kiv?
Oh yeah, Alpha Kive or whatever. Yeah.
Alpha Kive. Thank you.
This is a resource that I've been playing around with. So yeah, it's this tool Alpha Kive. You can turn any preprint and archive into a conversation using Alpha Kive. And it's project sponsored by Archive Labs. So this is where they're kind of doing this, I guess, peer review of the preprints or commenting on the preprints. It's kind of a nice tool.
I mean, I been certainly early supporter of Fermat's library in terms of what they were doing, but like you said, is that the way in which you can kind of get a larger group to narrow down on the small set of papers that they're going to read and really digest, so that it can become food for discussion, or springboards to new research initiatives. You know what I mean? Anyway, I'm curious how to. Well, yeah. I mean, I know there's there's the thing like NeuroStars. Right?
Yeah.
But NeuroStars, it seems like it's more textboard.
Yeah, it's like how to sort of debug.
Yeah, which does seem like what it was intended.
Right.
But it's like, I kind of feel, I don't know if that community exists, or because I mean, to some degree, I learn a lot from following like a couple of mailing lists in my field.
Yeah.
But it's not where people post their papers, or unless their paper comes up in the context of some analysis question or something like that.
Yeah.
It's not so much like, hey, everybody, you should read this. Anyway, I think that might also just be not being in a lab that's got a particular kind of funding focus, where obviously, again, there's a real need to stay to be familiar with the 5 to 10 other labs that kind of are doing that work. You know what I mean?
Yeah.
Anyway.
Yeah. So we'll move on. But interesting stuff. So the last thing I wanted to talk about this year in our meetings is this collection of papers on embodied and evolutionary approaches to morphology. So we've talked about sort of morphology as part of this embodied cognition. So we have the bodies of agents, and we talk about the brains of the agents, and we talked about morphological computation, which is how that morphology is organized to support cognition in the internal model of the agent, so the brains of the agent, and how it relates to the environment. So the three papers, I'm not going to go through them deeply, but I'm going to talk about them kind of in sequence. So the first paper is this new preprint. This is from Michael Levin's group and Leo Pio Lopez, who's, I think, driving the work on this particular aspect of Anthrobots, which is the morphological, behavioral, and transcriptomic life cycle of Anthrobots. So if you remember that, the Xenobots, which is like this, it's this Xenopus embryo that's sort of a robot. They're incorporating the cells into a robot, and they're using that as an autonomous agent. This is what they call Anthrobots, where they're using cultured adult human airway epithelial cells, and these cells become self-motile and acquire neural repair capabilities without exogenous genetic circuits or inorganic scaffolds. So they're these bots that are based on a human type cell that's in culture, and they're trying to engineer the cell to sort of engage in this sort of, motile behavior, the self-repair, and so forth. So that's what they're running. That's why they call it Anthrobot instead of Xenobot. So fascinating aspects of morphogenetic and behavioral plasticity of living material are revealed by novel constructs that self-assemble from genetically wild-type cells. Anthrobots, in this case, arise from cultured adult human airway epithelial cells, developing, becoming self-motile, and acquiring neural repair capabilities without exogenous genetic circuits or inorganic scaffolds. So they basically develop in this niche where there are these epithelial cells, they become self-motile, and then they become, they have the sort of plasticity where they can repair themselves, and there are no exogenous genetic circuits or scaffolds needed. So progress in bioengineering and regenerative medicine depends on developing a predictive understanding of collective cell behavior in novel circumstances. So, if we want to do bioengineering, we want to do regenerative medicine, we want to guide cells to a target, we want to do bioengineering where cells have to be sort of precise in where they go. We don't want to, sometimes we can have the luxury of building scaffolds into things, but, that's, sometimes we want to have things incorporated into a scaffold. We want to have greater control in bioengineering applications. So we want to develop sort of this autonomous behavior in cells. We want to do this from actual living cells instead of kind of engineering a solution that's inorganic. Towards that end, here we quantitatively characterize a number of life cycle properties of Anthrobots, including their morphogenesis, maturation, and demise. So they look at the life cycle of these cells, they look at the morphogenesis of the cells into this specialized cell type, they look at the maturation of the cell type, and then the demise or the apoptosis of the cell type. So, this is a differentiated cell. It will divide for a certain number of divisions, and then the cells will go apoptotic. So there's this life cycle that we have to be aware of. We uncover a self-healing capacity and a remarkable reduction of epigenetic age upon morphogenesis. Transcriptomic analysis revealed that assembling into Anthrobots drives a massive remodeling of gene expression relative to their cellular source, including several embryonic patterning genes, and a shift towards more evolutionarily ancient gene expression. So these epithelial cells sort of move around together in this sort of colony, and when cells are in a colony, they behave differently than when they're isolated. And so we want to understand where they, like if they come from a certain set of population of cells and they move to a new location and they integrate with another set of cells, how does that affect gene expression? How does that affect the further sort of morphology of the cell, the maturation of the cell, and so forth? So there's a lot of stuff that we need to know about these, and this paper is basically characterizing these Anthrobots. These data reveal new aspects of engineered multicellular configurations, in which wild-type adult human cells self-assemble into active living constructs with its own distinct transcriptome, morphogenesis, and life history. So they talk about Anthrobots, which are the first fully cellular human-derived non-embryonic biobots. They're self-constructing. They're considered organoids. Actually, I'm sorry. So the origin of these cells are, at least what they're characterizing, are these airway organoids. And these organoids are three-dimensional structures, of course, that grow in vitro that mimic the architecture and function of the human airway. So there's these human airway epithelial cells that are grown in an organoid. These organoids can derive from multiple sources, including stem cells, normal human bronchial epithelial cells, and human donors. Airway organoids are commonly characterized by their ability to replicate the cellular diversity of the airway epithelium, including ciliated cells, goblet cells, and basal cells. So a number of cell types. They exhibit key functional properties of the airway, such as mucus production, ciliary beating, in response to external stimuli. And so what they want here are these ciliated synthetic biological constructs. They want to understand those, so they want to know how those cells integrate into that structure. The main difference between Anthrobots and airway organoid is that while the primary goal of the organoids is to recapitulate the native tissue architecture and physiology, Anthrobots and other biobots demonstrate new living forms and function that is not explored by evolution but is useful for engineering purposes. They are used not only to make useful synthetic living machines, but also to explore motility-enabled behavior that reveals plasticity and emergent features not apparent in organoid assays. So this is a little bit about the Anthrobots. This figure 1, the caption is Anthrobots have different modes of self-construction that may account for emergent morphotypes. So they kind of show the different fates of the Anthrobot progenitor cell, which can be dormant, what they call a merger expander, and then what they call a monoclonal expander. So they kind of show in this, this is the PCA, where they show these three different sort of fates of the progenitor cell. So the progenitor cell is here, and in using a PCA, they can see that they have these three groups, these three axes of variation, these three sort of groups on PC1 versus PC2. And then here they have these different, they have basically the bot area, the number of events detected, and the variation and change of overall bot area. So these bots are collectives of cells that are in these different groups, and they're showing how this bot grows over time. And so then they kind of talk about a little bit about this. They show some of the staining of these bots and how they sort of form. Then they say Anthrobots exhibit a highly altered, more ancient transcriptome, including embryonic patterning genes. And they show some characterization here where they show that early stage Anthrobots display onset markers of embryonic development, that the progenitor cells are located in this part of this PC1 versus PC2 space, and then the day zero bots are down here. So clearly the day zero bots are different than the progenitor cells, which are shown in this red here, these red dots, and then the day zero bots are these assembloids that kind of form from the cells. And so this is the morphology of the bot. Anthrobots display different behaviors during the eversion process, which is this sort of eversion which brings the cilia into an outward-facing orientation. So this is like kind of analogous to an embryonic movement, like you would see like in invagination and evagination. So it's where it's folding in and out. Eversion is where the cilia become outward-facing. So they kind of characterize this process here. Anthrobots display different behaviors during the eversion process. And they show that here. Late-stage anthrobots display markers of maturing embryonic development. So this is where you see maturity in embryonic development that is, or you see anthrobots as they age displaying markers of maturing embryonic development. So this is where basically your assembloid is mimicking embryonic development in different ways. Anthrobots undergo a second massive transcriptomic rearrangement during their maturation. So the first transcriptomic rearrangement is when they sort of form these anthrobots, and the second rearrangement is as they mature. So there are a bunch of key genes that we can look at that involve mesoderm, involve ectoderm formation, and involve sort of endoderm-associated genes. So all three layers of the embryo are represented here. And so then in figure 4, they characterize late-stage anthrobots and how they display markers of maturing embryonic development. They show key transcripts here and how they change from your day 0 bots to your day 10 bots. So this is a 10-day sequence. And of course, in A, you can see that there's a colored representation of the anthrobots at day 0 and day 10. And you can see that in the stain, that there are differences here. There's also this change, full detection change in day 10 versus, up regulation, down regulation in day 10. And then in the PC space, PC1 versus PC2, the day 0 bots are down here. Remember, the precursors are up here in the space, day 0 bots are here, and day 10 bots are out here. So they change their position, especially in PC2, they sort of revert back to this sort of the level of the precursor cells, but PC1, they move further to the right. So that's, and so finally, anthrobots display robust behavioral longevity and reversal of epigenetic age. As they age, we get this behavioral longevity, and we get this sort of these mature anthrobots. And so they start to, they start to change in terms of their population size. Anthrobots approaching weeks 4 to 8 in their life cycle experience visible structural degradation. However, the frequency of motile behavior among intact bots at any given time remains steady, indicating bots do not cease their movement as they age, but only stop when they die. We found no evidence of an increasing elderly, fully immobile phase of life. So as the anthrobots age, cells die off, they become smaller. They still move around though, and they experience just sort of a mature phenotype. So that's really interesting. That's this Anthrobot approach to looking at morphology and, morphologies from cell collectives. The second paper is on embodied computational evolution, a model for investigating randomness and the evolution of morphological complexity. So this is by a pair of computer scientists, and there's some robotics research in here as well. So this is kind of proposing this approach called embodied computational evolution. So I'll read the abstract, and we can maybe go to the next paper in the interest of time. This is in the journal Integrative Organismal Biology. So this is, kind of focusing on biology instead of but it's also bringing computer science into the mix here. So the abstract reads, for an integrated understanding of how evolutionary dynamics operate in parallel on multiple levels, computational models can enable investigations that would be otherwise infeasible or impossible. We present one modeling framework, embodied computational evolution, or ECE, and employ it to investigate how two types of randomness, genetic and developmental, drive the evolution of morphological complexity. So you have this genetic component and a developmental component. So this is kind of like what we were dealing with in the first paper. We have gene expression, we have genetic randomness, we have developmental randomness, which they'll describe in the paper. But basically, those two things together, sort of the genetic aspect, and then, of course, in the assembloids, we have this developmental randomness that occurred from these cells assembling into these collectives. With these two types of randomness implemented as germline mutation and transcription error, in this case, they're defining germline mutation and transcription errors, these two sources of randomness, with rates varied in a 11 by 11 factorial experimental design. We tested two related hypotheses. So hypothesis one was randomness in the gene expression process alters the direct impact of selection on populations. And then hypothesis two is selection on locomotor performance targets, morphological complexity. So this is where you're looking at the gene transcription process, sort of altering selection, and then selection on locomotor performance targets, morphological complexity. So I guess they have these agents that they're doing this with. The experiment consists of 121 conditions. In each condition, 9 starting phenotypic populations developed from different randomly generated genomic populations of 60 individuals. Each of the resulting 1,089 phenotypic populations evolved over 100 generations, with the autonomous self-propelled individuals under directional selection for enhanced locomotor performance. As encoded by their genome, individuals had heritable morphological traits, including the numbers of segments, sensors, neurons, and connections between sensors and motorized joints that they activated. An individual's morphological complexity was measured by 3 different metrics derived from counts of the body parts. In support of hypothesis 1, variations in the rate of randomness in the gene transcription process varied the dynamics of selection. In support of hypothesis 2, the morphological complexity of populations evolved adaptively. So this is the ECE approach here, where they show this process. We have the embodied process, which is where we have, it's kind of a later process. We'll start with the genomic process, where we have this deterministic aspect of selection. We have the parents. We have mutation, which is genetic error. And we have reproduction. Then we have development, which is this mapping error, mapping the genes to phenotype. And then you have behavior, which is part of this embodied process in orange. And then you get back to this deterministic aspect of selection. So ECE is an agent-based modeling framework that incorporates the entire life cycle of each individual in a population of those individuals. So you have this population of agents that you're selecting on. You're mutating their genome. You're mapping the genotype to phenotype or to behavior. And there's a mapping error in development. There's a genetic error in the mutation process that then leads to this, some variation in the population that you can select upon. So embodied processes include the development of the individual and its behavior in an environment. Behavioral differences among the individuals in a population are used deterministically to select the parents in the population of reproduction. Genomic processes may include the mutation of the genome that is used in reproduction. The genome is expressed in the developmental process. Random errors can, in principle, be introduced at any place in this life cycle. So we have this approach to embodiment. In this paper, we commonly call these individuals biorobots because they are embodied models built to test biological hypotheses in the manner of Webb's biorobotic paradigm. This is Webb 2001. And so that's basically what we're doing. We're doing a very similar thing what we did with Anthrobots in the first paper. Basically, their agents have genomes with genetic interactions, development of fixed and or variable rules, physically embodied morphology with sensory motor circuits that drive self-propulsive body reconfigurations, and behavior that is autonomously generated by the organism interacting with its physical environment. This third paper then is Evolutionary Robotics as a Modeling Tool in Evolutionary Biology. And again, we have this robotics approach, but this time it's sort of as applied to evolutionary biology, not biological constructs or agents that are supposed to be biological constructs. So this is, this is from Frontiers in Robotics and AI. I'll just go through the abstract and we'll finish up for today. So the use of evolutionary robotic systems to model aspects of evolutionary biology is well established. This is sort of a lot of this work on morphological computation comes from evolutionary robotics, not organismal biology. And as such, we can, more easily quantify the phenotype, of course. But then there are drawbacks to this as well, such as, how do we actually apply it to biological systems and this sort of vertical organization that we see in biology. So, we've done this, a lot of things with looking at evolutionary biology using evolutionary robotic systems. Yet few studies have asked the question, what kind of model is an evolutionary robotic system? And so this paper seeks to address that question in several ways. First, it is addressed by applying a structured model description developed for physical robot models and animal sensory motor systems. Then by outlining the strengths and limitations of evolutionary robotics for modeling evolutionary biology. And then finally by considering the deepest questions in evolution and which of them might feasibly be modeled by evolutionary robotics. So we're looking at structured models, which describes sort of physical robotic models of animal sensory motor systems. So we're interested in looking at animal sensory motor systems, how they perform. We can do things with the robots that we couldn't do with animals. We can look at their structure. We can look at their function. We can also evolve them in a computer and see what, how that process would proceed. Then we outline the strengths and limitations of evolutionary robotics for modeling evolutionary biology, which we talked about before. And then we consider the deepest questions in evolution, which I guess are, how do these agents evolve? What are their common ancestry, points of common ancestry, etc.? What is their phylogeny? And also you could answer questions in Evo-Devo or the evolution and development. This paper concludes that although evolutionary robotics faces serious limitations in exploring deeper questions in evolutionary biology, as one might expect, because it's hard to sort of get a handle on very deep phylogeny and some of the questions related to that. In other words, we can model extant organisms and we can understand that because we can build good representations of that, but it's hard to build good representations of things that we don't really have good extant models of. It's kind of speculative at that point, but it's something that we can use as a tool. It's particularly the bottom-up approach to modeling populations of evolving phenotypes and their embodied interactions. So we want to understand not just single sort of idealized agents in their design, but we want to understand the set of population level. We want to know the kind of variation that might be created in sensory motor systems, or the variation that might be inherent in a population of phenotypes that do things functionally. We can do this at the bottom-up approach. We can start with the genotype, work our way up to the phenotype, and have populations that exhibit mutation, exhibit selection, exhibit genetic drift, exhibit functional variation, structural variation, and the like. We can have phenotypes that we can measure, and we can have phenotypes that evolve. And this all involves a number of embodied interactions. And so, if we want to test hypotheses about embodiment, we have to kind of give ourselves, not just agents that are idealized, but agents that are sort of, that have variation. And we want to understand how these phenotypes evolve, because part of, I think, embodiment is knowing how a phenotype got there. embodiment, if the embodiment hypothesis is true, it would suggest that function and structure kind of co-evolve, that you can't really have refined function without refined structure and vice versa. The brain isn't just in a vat and it doesn't like optimize itself and fit into any old phenotype. The phenotype co-evolves with the brain, with the controller. And so you'll see that, but also, there's a population of different possibilities that could lead to some evolutionary trajectory. So that's something we have to keep in mind. And then the developmental trajectory. How do these things kind of get coordinated in development? How do they come online? And then how does that affect the evolution of these systems, the phenotype and the genotype and the controller, the behavioral controller? And how do those all fit together? And how do they become sort of optimized, or what some people call a good fit? And so that's where, that's where we can use these kind of evolutionary robotics models. That's where the power lies in simulation and some of the analogies that we raised from this work. Okay. So that's all for that set of papers. Do we have any comments or questions on that?
Well, yeah, I mean, I love this topic.
Yeah.
I just saw Dawkins talking about population genetics, and I felt like one of the problems of kind of understanding his nuances is like, that you kind of need to be familiar with the simulations.
Yeah.
Like it seems like it's an amenable, discussion topic to, it's an open area. You can just walk in and get all the points. But, I think, I really like this, the second paper, I got thinking about that. And, just that's a really interesting way of seeing, Bongard's papers too, of course, right? Yeah. Not the first one, right, which is a whole other, set of things, but like his kind of what he's known for, right? Yeah. Or, he had one of the first papers, I think, I think his Bongard's like first evolutionary robotics paper was 20 plus years ago.
Yeah. That's
yeah. And I didn't hear about it until it started hitting like PNAS or something.
Yeah.
And anyway, but to your point, right? That like these are not easy things to just kind of armchair think about. These are it's actually really important to see these embodied, multi-scale simulations for what insights they give for, again, like, I'm just, it's quickly fell down a rabbit hole of following up, some references and seeing that, these are actually early Josh Bongard papers.
Yeah.
Bongard and somebody else, Randall Beard.
Okay.
Yeah.
Beard.
Yeah. Like as another person. Yeah. And anyway, so these are really interesting. It's kind of, well, but of course, the Anthrobots is another whole, another, because like there you see the difference between the robotics that we have, right? Which is like our crude way of making things, our brittle way of making things, and dealing with agential matter. Right. Because, I mean, really I mean, this is like completely colored by seeing, Michael Levin and Bongard to some degree, but Michael Levin all over the internet, talking about this work, but like, that nobody knew that this matter would do this.
Yeah.
It's if we release it from the normal local environmental context, if we release it from its collective bonds, will it, like you said, like undergo a kind of embryonic reconfiguration,
right?
that generates a machine that we've never seen before, and and what that suggests, and like I don't know how much these papers could get in, again, like you're not dealing with that with these mechanical robots, because of course they are very limited and totally passive materials, right? Anyway, it's super interesting. So I'm going to meet in a half an hour with Neurotech of Berkeley to talk about a second project that's very much dish brain and or beyond, right? And but the Anthrobots make me think about how some of Levin's experiments are actually quite accessible,
right?
Like with like this isn't Allen Institute required, well, of course it's funny that he is, of course, the Allen Institute as well, but but this doesn't require big capital costs to do these kinds of experiments, right?
Yeah.
And I wonder, I'm it's like you've got all the problems of cell culture, mammalian cell culture, right? With certainly kind of dish brain and or anything that's going organoid, as well as like there's this very day-to-day maintenance, right? So it's just like if you get it started and it's working, right, then it requires a very constant maintenance regime, and to even get the things that you're going to want to get, it's just a good reminder that there are other kind of biology experiments that I think are really interesting, that speak kind of to the collective what we like about neurons are not neurons, but there's actually networks of neurons.
Right. Yeah.
Right. and I like that Levin's shown us that you don't even need neurons.
Right.
This is what all cells are doing.
Right.
And we aren't just thinking about it in the right way. And are there perhaps other, so it's just food for thought for this discussion with, like, how much time do you guys have for what is ostensibly an extracurricular activity?
Yeah.
Like whether we should put our kind of eggs in a basket that requires such kind of consistent daily work, or if it should be something more focused, but shorter period, but kind of more interesting, perhaps more interesting demonstration or something like that. Anyway, as usual, Levin's work just continues to inspire.
Yeah.
And, for sure. Anyway, so this is it for the holidays?
Yeah, that's it.
Okay.
And if you'd like to meet during the holidays sometime about anything.
Yeah, I wanted to know if we could, if it made sense, and perhaps with whose support it would make sense to do something, either NeuroFedora or I would still really like to do something for NeuroFedora around the Insight toolkit.
Okay.
So this is big National Library of Medicine, registration segmentation toolkit, and it's a massive C++ project, well documented, well tested, that is actually used by a lot of other tools.
Okay.
Now, so this is kind of a software engineering project, and I was wondering whether to reach out to a group that focuses on like the software engineering for medical imaging.
Yeah.
As well as like to reach out to somebody from Kitware, because I've definitely got some good Kitware contacts. Kitware was the kind of software development group that was given the contract to develop this. It's a public project in the sense it was under National Library of Medicine, but Kitware, because Kitware is already kind of known for its big open source scientific libraries like VTK.
Okay.
And of course now so many more projects use CMake now than say, Automaker, any of the other kind of old tools. CMake is a Kitware project.
Okay.
So CMake was developed to build VTK and ITK. and to deal with very disparate platforms, so it's like make it so you can build on Windows, you can build on Linux, build on Mac, and so I would suggest, like, interested in using these tools, using the most recent versions of these tools. And then this is a project that would be the first goal. Like
Okay.
let's build Ant toolkit with these, because but that's already building, right? That's very simple. It'll be like an easy kind of win, and it'll demonstrate something. Then here's another toolkit that uses ITK and VTK and it's ITK Snap. And it's a limited GUI toolkit, but again, it's got some really cool features, and it would be a great addition. And basically working up in terms of complexity to 3D slicer, and kind of say, if the person doesn't hit, if you don't manage to get all of 3D Slicer working on Fedora, that's not a failure. But there could be goals that we'd hope to get to, and that would be kind of the reach goal.
Okay.
I don't know how you kind of frame that for mentees in terms of it would be great, it'd be great for you to do this. I mean, do you give them limited goals that you're pretty sure that they can hit? Or do you say hey, this is the goal, but it's okay to not reach that. We'll just say, you know, after 10 weeks, like these are the reasons, you know, we learned this about
Yeah.
each of them.
Yeah, they have to develop a schedule or you know, so they have to like kind of hit. I usually don't get, I usually don't have projects that are that like sort of goal oriented. Like I usually have them develop something on their own, but then, you know, I have them do their schedule and say, okay, you're going to hit these goals. And if you don't hit them, you have alternate, like you can leave some room in the schedule so that, you know, you hit these goals by these weeks. But if you don't, then you have these alternate goals. And you could actually put it in the description and say, we would like to do these things, but, you know, you can also kind of, you can build your schedule so that you only reach the first goal. And it really kind of gives them this, and it's up to the person reviewing the applications and to say, is this a reasonable way to reach this goal or, you know, not? And, you know, it's largely up to the person proposing when they propose, they have to propose a solution to the problem and how they're going to achieve those goals in this amount of time. So we'd say, like, this is what we want to accomplish with this toolkit. You know, you're going to build this and get to this point at, I guess, 12 to 14 weeks. If you can't do that, then this is a stretch goal. This is a goal that we really want to hit. And then they propose how they're going to do this. You have to evaluate whether that's a reasonable solution, whether they can hit those goals. And then it's up to them if they can do it. And hopefully they can do it. A lot of people can because they're usually pretty motivated. But, you know, that's also up to the mentor to say, you know, we need to sort of find the best path to getting this done in this amount of time. It's a really tough skill to master because, you know, there are a lot of moving parts and they're going to encounter, you know, blockers and other types of things. And so it's really kind of, you know, we have to kind of, I guess, give them a good basis for sort of setting up that schedule. That's really what we need to do in the in the call for involvement.
Mm. So I'm just trying to think about like ways to kind of inform that, and like, you know, like maybe, you know, reaching out to some of the, like the 3D slicer community and saying like, hey, what have been the big stumbling blocks for people to do this? Right. You know, why haven't you taken this on? Like, are there particular stoppers? Yeah. And then be able to say more about, okay.
Basically, we want to leave it somewhat open-ended and not too open-ended, and because we don't want them to go down a blind alley and then not be able to.
Absolutely. I mean, that's why, you know, and it would be like, I mean, I would definitely want to have, you know, I mean, again, like I'm happy to pitch this just to Kitware and say, you know, are you guys interested in kind of sponsoring this? Yeah. Like, you know, okay. Well, I'll see what I can. See what I can learn, and happy to talk over the holidays.
Yeah. Set up a time. We'll do that.
Yeah. Okay.
All right.
Well,
thanks.
Yeah. Happy Christmas, as they say in the UK.
Okay. Happy holidays to you too.
Yeah.
All right. Hope to touch base.
We'll talk later.
All right. Talk later. Bye.
Cheers.
Bye.